Dear students, this is Dr. Heba Ghannoum, lecturer of audiovestibular medicine, and this is our first otology lecture, Anatomy of the Ear and Physiology of Hearing. Anatomy of the ear. The ear is composed of outer ear, middle ear, and inner ear. The outer ear is composed of three structures: the auricle or the pinna, the external auditory canal, and the drum, whereas the middle ear is composed of the middle ear cavity, anteriorly the Eustachian tube, and posteriorly the mastoid air cells. All three together, these three together, we call them the middle ear cleft. The inner ear is composed of the bony labyrinth. If we open or break this bony labyrinth, inside we'll find the membranous labyrinth and the sensory end organs. Now we start to explain each structure in details. We start with the auricle or the pinna. This is a piece of cartilage. On its lateral surface, multiple ridges and depression. We start with the uppermost rolled ridge called the helix. Then opposing to the helix, a Y-shaped ridge called the antihelix. In between the two crura of the antihelix, there is a fossa called a triangular fossa. Just beneath the helix, this is called the scaphoid fossa. Anterior and inferior to the antihelix is the concha, this part, and then the external auditory, the meatus, which is the opening of the external auditory meatus, covered by a protective ridge here called the tragus. Opposing to the tragus is another ridge called the antitragus. This is the lobule, the only portion or part of the pinna which is devoid of any cartilage. It's made only of fat covered with skin. So we said here the helix, the antihelix, triangular fossa, and scaphoid fossa, and here the concha is divided with the cross of helix into two parts. This part here is called the Simba concha. Simba concha is an important landmark, clinical landmark, because if you press on the Simba concha and there is tenderness, this signifies the presence of inflammation in the mastoid antrum, mastoiditis. The external auditory meatus, or the external auditory canal, it extends between the auricle and the tympanic membrane, and it measures 24 mm in length. It's divided into two parts, the outer 1/3 and the inner 2/3. What are the differences between the two parts? The outer 1/3 is cartilaginous, whereas the inner 2/3 is bony in nature. The outer 1/3 is covered with thick skin containing the hair follicles and the sebaceous and the ceruminous glands, whereas the inner 2/3 is lined with thin skin devoid of any hair follicles or glands. The third difference between the two parts is the direction of the canal. The outer 1/3 is directed upwards, backwards, and medially, whereas the inner 2/3 is directed downwards, forwards, and medially. What about the anatomy of the tympanic membrane? It's oval in shape, almost 9 × 8 mm in diameter. It's pearly white in color, semi-translucent. It's concave when we see it from the lateral view. And the lower 2/3 of the tympanic membrane is surrounded by a thickened fibrous ring called the tympanic annulus. The position of the tympanic membrane is placed in an oblique position, 55 degrees, at an angle of 55 degrees with the floor of the external auditory canal, so that the anterior and the inferior walls of the external auditory canal are longer than the posterior and the superior walls. What are the landmarks on the tympanic membrane? Here, the anterior malleolar fold, the lateral process, the handle of Meatus, and the posterior malleolar fold. Here, around the lower 2/3 is the thickened fibrous ring, what we call the tympanic annulus, and here is the cone of light. There is another landmark here. This is the most depressed part on the tympanic membrane. It's called the umbo at the end of the handle of Meatus. The anterior and the posterior malleolar folds divide the tympanic membrane into two parts. Upper part, we call it the pars flaccida, and the lower part, we call it the pars tensa. What's the difference between the pars flaccida and the pars tensa? The pars flaccida is the flaccid part that has no fibrous layer and is not surrounded by the tympanic annulus, whereas the pars tensa is the lower tense part that has a fibrous layer and is surrounded by the tympanic annulus. طيب, what do you think? Which part is more liable to tympanic membrane rupture upon receiving a slap on the face, as an example? Definitely the pars tensa, because it will resist moving back and forth because of the tense fibrous layer. What are the layers of the tympanic membrane? It consists of 3 layers. Outermost layer, or the lateral skin layer, that's continuous with the skin lining of the external auditory canal. The middle fibrous layer, this fibrous layer is, as we previously said, it's well developed in the pars tensa and it's absent in the pars flaccida. The handle of Meatus is embedded in the middle fibrous layer. Then the third layer is the inner mucosal layer, which is a continuation of the mucosal lining of the middle ear. Okay, what's the thickness of the tympanic membrane? And is it thicker in adults or in children? What do you think? The drum is 0.1 millimeter in thickness in adults, and it's definitely thicker in children. Why is it thicker in children? Because they have a narrower external auditory canal, and as they grow, it widens, and so the tympanic membrane is more stretched and thus gets thinner as they grow. Does this have any clinical relevance? Yes, it does, because a thicker drum will resist bulging and will resist perforation. So this makes children more liable to the cases of complication of acute otitis media than adults. So we finished the outer ear and we start with the middle ear. The middle ear cleft is formed of, as we said, is formed of 3 parts. The middle ear cavity. The middle ear cavity is connected anteriorly through the Eustachian tube to the nasopharynx, and posteriorly through the adductus ad antrum to the mastoid air cells. So what are the 3 components of the middle ear cleft? The middle ear cavity, Eustachian tube, and the mastoid air cells. The middle ear cavity, it lies between the external ear and the inner ear. It is a 6-walled cavity. ازاي 6-walled? It has a floor, a roof, anterior wall, posterior wall, medial and lateral walls. يبقى 6-walled cavity divided into 3 compartments. If you take a look at the diagram, you will see 2 lines extending from the tympanic membrane. They divide the middle ear cavity into 3 compartments, these 2 lines here. So 3 compartments. The uppermost compartment, we call it the epitympanum or the attic. It lies above the tympanic membrane and it's continuous with the mastoid antrum posteriorly by the adductus ad antrum. The epitympanum contains most of the ossicles, as you can see from the diagram. The second part is called the mesotympanum, and this is the part facing opposite to the tympanic membrane. And the third part is called hypotympanum, and this lies below the tympanic membrane. Now this is very important, walls of the middle ear cavity. As we mentioned earlier, that the middle ear cavity is a 6-walled cavity. I want you first to imagine the diagram. Here we removed the lateral wall, the tympanic membrane, and we're looking from the inside. So we have here the floor, the roof, the anterior wall, the posterior wall, the medial wall here inside, and we remove the lateral wall. Okay, let me erase this. Let's start with the roof of the middle ear cavity. The roof is formed of a thin plate of bone called tegmen tympani, and it separates the middle ear cavity from the temporal lobe of the brain. What about the floor? The floor is even a thinner plate of bone, very thin plate of bone that separates the middle ear cavity from the superior jugular bulb, and sometimes it is deficient and the patient complains from what we call pulsatile tinnitus. You will know about this later. Okay, so we finished the roof of the middle ear cavity and the floor. We move on to the anterior wall of the middle ear cavity. The anterior wall has 3 canals: a canal for tensor tympani muscle, the Eustachian tube, and the carotid canal for the internal carotid artery. What about the posterior wall? The posterior wall has 3 features. The first one is the adductus ad antrum. This one, it connects the attic to the mastoid antrum. The second feature is the pyramid for stapedius muscle, and the stapedius tendon emerges from the tip of this pyramid and ends at the stapes. The third feature is the vertical part of the facial nerve in its bony canal. So these are the 3 features of the posterior wall: addutus ad antrum, the pyramid for the stapedius muscle, and the vertical part of the facial nerve in its bony canal. What about the lateral wall? The lateral wall of the middle ear cavity, it has the tympanic membrane to separate it from the outer ear. Also, in addition to the tympanic membrane, it has bone from above and bone from below. What about the medial wall? The medial wall is the most important. The medial wall shows, first of all, the promontory. Here, this bulge, this central rounded bulge called the promontory. What is the promontory? This is a bulge caused by the first turn of the cochlea. And the medial wall separates the middle ear cavity from the inner ear. So the promontory is a central rounded bulge that is caused by the first turn of the cochlea. Above the promontory there is an opening, and below the promontory here there is another opening. So we have 2 windows: the oval window above and behind the promontory, and the round window below and behind the promontory. Above and in front the promontory we have a structure, a bony structure that I mentioned earlier called the processus cochleariformis. The last feature of the medial wall is the horizontal part here of the facial nerve. The horizontal part of the facial nerve in its bony canal above the promontory and the oval window. Also, there is another one final feature in the middle, in the medial wall, here in between, in this area, in between the oval window and the round window, there is a depression called the sinus tympani. What are the contents of the middle ear cavity? Air coming from the Eustachian tube, and then we have 3 bones or ossicles, 2 muscles, and 2 nerves. The 3 bones or the ossicles, malleus, incus, and stapes. The 2 muscles, tensor tympani and the stapedius. Tensor tympani, as we mentioned earlier, entering from the anterior wall, and the stapedius from the posterior wall. And the 2 nerves, the chorda tympani muscle, the chorda tympani nerve, and the tympanic branch of the glossopharyngeal, the one we saw arborizing on the promontory. This is called also Jacobson nerve. The Eustachian tube is 36 millimeter in length. If you take a look at the diagram, please. Here is the Eustachian tube. It connects the middle ear with the nasopharynx. It's directed downwards, forwards, and medially, composed of 2 parts. The lateral 1/3 here, close to the middle ear cavity, is bony, whereas the medial 2/3 is cartilaginous. And it is composed of, the medial 2/3 is composed of incomplete C-shaped cartilage, because normally the Eustachian tube is usually closed, is always closed, and it opens during swallowing or yawning by the action of the palatal muscles, the levator palati and the tensor palati muscles. What is the function of the Eustachian tube? It has 2 functions. Ventilation of the middle ear cavity. It equalizes the pressure inside the middle ear with the atmospheric pressure, and so this allows the tympanic membrane to move freely in response to sound. And the second function of the Eustachian tube is middle ear drainage. In this slide, we can see the difference between the Eustachian tube in adults versus Eustachian tube in children. Eustachian tube in children, as you can see from the photos, is shorter in children, wider, and is more horizontal. And that's why otitis media is more common in children than in adults. Mastoid air cells. These are small air-filled bony cavities. They lie within the mastoid process of the temporal bone. The mastoid process is pyramidal in shape and the tip is directed downwards. The largest air cell هي الماstoid antrum, and it communicates with the attic through the additus ad antrum. Other groups of cells exist, and they are named according to their location or their site. Behind the mastoid antrum there is the periantral cells. Here this is the sigmoid sinus, and so this group is called the perisinus cells. At the tip we find the tip cells. Near the facial nerve, behind the facial nerve, retrofacial, and another group in between the dura and the sigmoid sinus, so it's called sinodural cells. Now that we are done with the external ear with its structures, the auricle, the external auditory canal and the drum, and the middle ear cleft with its 3 structures, the middle ear cavity, the Eustachian tube anteriorly, and the mastoid air cells posteriorly, we move on to the inner ear. This part we zoom in and magnify it to get this image. The inner ear is composed of 2 parts, the bony labyrinth and the membranous labyrinth. if we open the bony labyrinth, we'll find the membranous labyrinth inside. The bony labyrinth is composed of three structures: cochlea, vestibule, and semicircular canals. The cochlea, this snail-shaped structure, is composed of almost 2.5 turns, with the basal turn towards nearer to the vestibule and the apex far away. This is the vestibule in between the cochlea and the three semicircular canals. The three semicircular canals, the anterior, the posterior, and the lateral canal or the horizontal canal. Each of them is open, has two openings in the vestibule, except for the anterior and posterior. They form a common cross and they open with one opening. So instead of having 6 openings in the vestibule, we have only 5 because of the common cross between the anterior and the posterior semicircular canals. The bony labyrinth is filled with perilymph. The perilymph is similar in its composition to the extracellular fluid. What does this mean? It means that it is low in potassium and high in sodium. If we break or open the bony labyrinth, we'll find inside it the membranous labyrinth. The membranous labyrinth is composed of the cochlear duct lying inside the cochlea. Inside the vestibule, we'll find 2 sacs or 2 chambers, the saccule and the utricle. And inside the semicircular canals, we'll find the 3 semicircular ducts. In addition to a 4th structure, the endolymphatic duct and the endolymphatic sac, this portion. The membranous labyrinth is filled with a fluid called endolymph. This endolymph is similar in structure to the intracellular fluids. This means that it is high in potassium and low in sodium. If we take a cross section in one of the turns of the cochlea here, so these are supposed to be the 2.5 turns of the cochlea, and here the vestibule and the 3 semicircular canals, as we earlier said. In this diagram, I want you to imagine we are taking a cross section in one of the turns of the cochlea, and here is what we get. The cochlea is divided into 3 compartments by 2 membranes. So the 3 compartments are the scala vestibuli, the scala media, and the scala tympani. The 2 membranes that divide the cochlea into those 3 compartments, the first one is called Reisner's membrane. It divides the scala vestibuli it separates the scala vestibuli from the scala media. And then the basilar membrane, this one, it separates the scala media from the scala tympani. On top of the basilar membrane and inside the scala media, you will find the organ of Corti. What is the organ of Corti? This is the sensory organ of hearing. This is another cross section in one of the cochlear turns, showing the 3 compartments of the cochlea: scala vestibuli, scala media, and scala tympani. Scala vestibuli and scala media are filled with perilymph, and they communicate together at the apex, at the helicotrema. The scala media is filled with endolymph, and this is the stria vascularis, which produces the endolymph. So the production of the endolymph comes from the stria vascularis, and its drainage, if you remember earlier, we mentioned the endolymphatic duct and sac. The endolymphatic duct and sac are responsible for the drainage of the endolymph. Also in this diagram, the organ of Corti is very nicely represented. As you can see, it sits on the basilar membrane. It's composed of 1 row of inner hair cells, 3 rows of outer hair cells in addition to the supporting cells. The stereocilia above the hair cells are in contact with the tectorial membrane. This is the tectorial membrane. So it's very nicely represented. This is the organ of Corti, the sensory organ of hearing. In this diagram, we can also see the modulus, which is the axis around which the cochlea make its turns. It contains the spiral ganglia, which then emerges to form the auditory nerve fibers and the cochlear branch of the 8th cranial nerve. What are the sensory end organs of hearing and equilibrium? The sensory end organ of hearing lies inside the cochlear duct. As we mentioned it earlier in the last slide, it's called the organ of Corti, whereas the sensory end organs of equilibrium lies inside the semicircular canal and utricle ancicule, the vestibular apparatus. Inside the semicircular canal, it's called crista ampullaris, and this is responsible for angular acceleration or the angular movements, whereas inside the utricle ancicule, the macula is responsible for the linear acceleration. So what is the physiology of hearing? If we start with the external ear, sound reaches the external ear, the auricle. The auricle acts as a funnel that collects the sound and is responsible for localization of the sound as well. Sound is then transferred from the auricle through the external auditory canal to reach the tympanic membrane, causing its vibration. In the middle ear, sound will be conducted from the tympanic membrane to the oval window. The vibration of the tympanic membrane will be transferred to the ossicles, causing movement of the ossicles. Inside the middle ear, there is a process of amplification of sound, 22 times almost. This happens by two factors. The first factor is called the surface area factor, or the surface area ratio, which is the difference between the diameter of the tympanic membrane when compared to the diameter of the foot plate of stapes at the oval window. The second factor is the lever action of the ossicles. The lever action is also the ratio between the length of the handle of malleus and the long process of incus. So the total the overall amplification is the multiplication of the 17 times from the surface area ratio and the 1.3 from the lever action of the ossicles, and we get 22 times amplification inside the middle ear. Why is this amplification important? This is important to counteract the impedance mismatch that would happen in case the sound is transferred from air to fluid inside the inner ear. Also, a very important function of the middle ear is equalization of pressure on either side of the tympanic membrane through the Eustachian tube, so that the tympanic membrane can move freely and transmit the sound. So let's watch
sound
this video
entering the
to
ear
sum up what we have said so far.
The external auditory canal before striking the eardrum and causing it to vibrate. The eardrum is connected to the malleus, one of 3 small bones of the middle ear. Also called the hammer, it transmits sound vibrations to the incus, which passes them to the stapes. The stapes pushes in and out against the structure called the oval window. This action is passed on to the cochlea, a fluid-filled, snail-like structure that contains the organ of Corti, the organ for hearing. It consists of tiny hair cells that line the cochlea. These cells translate vibrations into electrical impulses that are carried to the brain by sensory nerves. In this cut view, you can see the organ of Corti with its 4 rows of hair cells. There is an inner row on the left and 3 outer rows on the right. Let's watch this process in action. First, the stapes rocks against the oval window. This transmits waves of sound through the cochlear fluid, sending the organ of Corti into motion. Fibers near the upper end of the cochlea resonate to lower frequency sound. Those near the oval window respond to higher frequencies.
So as we have seen in the previous video, the back and forth movement of the stapes, the piston-like movement, will create waves inside the inner ear fluid, the perilymph in the scala vestibuli. This will then be transferred to the scala tympani and cause the bulging of the round window. This is called the phase difference. This pressure will cause the vibration of the basilar membrane, up and down vibration of the basilar membrane. When the basilar membrane vibrates, as we have said earlier, the organ of Corti is located on top of the basilar membrane. The hair cells will move against the tectorial membrane. This movement of hair cells will develop a receptor potential that will lead to the generation of nerve impulses. These impulses are then passed to the cochlear branch of the 8th cranial nerve and then to the medulla. We have previously mentioned the functional structure of the organ of Corti, which is the sensory epithelium of the inner ear responsible for hearing. Again, it sits on top of the basilar membrane. The organ of Corti is composed of inner hair cells, outer hair cells, supporting cells, and the tectorial membrane. On top of the hair cells, we find the stereocilia that are attached to the tectorial membrane. The up and down movement or displacement of the basilar membrane cause the hair cells to move against the tectorial membrane. This movement of hair cells or stereocilia against the tectorial membrane cause the bending of the stereocilia and thus opening of the ion channels. What happens is the influx of potassium inside the ion channels, through the ion channels inside the hair cells, and this causes depolarization of hair cells and release of neurotransmitters. The release of neurotransmitters will then elicit a response in the primary auditory neurons. Now the central auditory nervous system or the central auditory pathway. You probably know this from physiology. Let's revise it quickly. The receptor is the organ of Corti inside the cochlea. First order neuron, the spiral ganglia. This will relay in the second order neuron in the dorsal and ventral cochlear nuclei. Then fibers will pass to the superior olivary nucleus on both sides and will ascend in the lateral lemniscus to the inferior colliculus, and then third order neuron, the medial geniculate body, and will end in the primary and secondary auditory cortex in the superior temporal gyrus. Physiology of equilibrium, I will not mention this in details right now. We need to know that what maintains the body balance is vestibular apparatus, vision, and proprioception. What is the vestibular apparatus? The vestibular apparatus is composed of the semicircular canals, 3 semicircular canals lying perpendicular at 90 degrees to each other, and the otolith organ inside the vestibule. The otolith organ is composed of 2 chambers, the saccule and the utricle. The vestibular apparatus, as we said, is composed of the semicircular canals, 3 semicircular canals lying perpendicular to each other, and the otolith organ. The otolith organ is the utricle and the saccule. At the end of each semicircular canal, there is a dilated end. We call it the ampulla. Inside the ampulla, the receptor called the crista ampullaris is located. The crista ampullaris is responsible for is the receptor of angular acceleration, responsible for detecting rotatory movements. Whereas inside the utricle and saccule you will find another receptor called the macula. The macula is the receptor for detecting the linear acceleration that is affected by gravity. You will know more details about the physiology of equilibrium and the mechanism of action of the crista ampullaris and the macula later on, but we'll stop here for now with my best wishes. Thank you.